you for that kind of introduction, Steve. I must say that a lot of the work that we're going to talk about today is joint work with Elias Chebovneto, who you'll hear from later in the day. So a little bit more about me. I came to Madison about 30 years ago, a little over 30 years ago, and I joined statistics and horticulture as part of the biometry team. Now, I don't have any background in horticulture except I grow a few plants here and there, and I've got peppers on my shirt. But it embedded me in the biological community, and I get most of my ideas as a statistician out of collaborations with biologists. And I got exposed to genetic ideas from Tom Osborne and other biologists, and got involved with brassica in some early QTL work. So that got me started, and I'll actually be referring to some of that work here. The website indicated here has all the slides from this this talk, and there's links to other talks that I've given. Also has collections of references with hot links to. I've tried to keep up a collection of all the QTL references that there have ever been. There's about 1,000 references, and they're all hot links, so you can get directly to the articles if you're interested. Okay, so we're talking about correlated traits, and we're talking about networks, and when we're talking about about biological systems, we measure gross phenotypes, we measure gross characteristics, such as what Mark was talking about, one of my biology collaborators, in terms of glucose and insulin levels. But we know that there's a bunch of other processes that are going on behind and over a period of time to get to whatever glucose level an individual mouse has at 10 weeks. So we have the central dogma of biology going from DNA to RNA to proteins, and lately Richard Janssen's been a leader in this area of taking this dogma and moving it into the gene mapping era. So we have location of SNPs up here, and then we have mRNA expression measured here, protein measured here, and then various other things, and then gross phenotypes down here. And you see some characteristics, like over here, are followed back through the molecular history. So you can find some interesting connections, some of them not so much. And some of the molecular measurements don't end up coming through as phenotypes. So there's a buffering going on, there's a lot of redundant processes going on, but this is something we're trying to uncover the biochemical basis of gross phenotypes. And we saw some molecular pathways the last couple days. I'll just pass through this. But all of the pathways that have been developed are actually available on this interactive website if you're interested in looking at it. KEGG pathways were mentioned. So I'm going to be talking about a systems genetics approach. We're bringing genetics in, and we're trying to use the genetic architecture, that is, what parts of the genome are responsible for differences in levels of expression, say. So a lot of the work up till now, with the exception of Mark's talk yesterday, had to do with having different conditions. You have animals in one condition versus another condition. What we're going to talk about here is where we use genetics as the basis of condition, and try to understand what regions of the genome are responsible for phenotypic differences. Okay. So there's a lot of detail about how you might want to drill into the system genetics. So I imagine a number of you haven't had a background in gene mapping, so I will point you to the expert, Carl Broman. He's got a book, Broman and Sen, which is available. He has a website where you can find all sorts of things. And then this is one of his talks, and I just want to go briefly to this talk here. So I'm just going across the internet to his talk. Rather than copy his slides, I thought I'd just use the slides directly. So here we're taking, the work I'm going to talk about, and that what Mark was talking about largely has to do with having experimental cross, in this case mice. So the mice have an advantage that we start from inbred strains that are completely identical, and so we can grow up a lot of them, and then study mouse as a model for humans. And the experimental situation is you start with two parents that are completely inbred, one of one type, one of another, so this would be like B6 and BTBR, and then you cross them to get an F1, and then in this case you back cross, and in the back cross one of the chromosomes is going to be identical to the first parent, and then the other one is going to be some sort of a mix. And horizontally looking across here, taking a particular line here, will correspond to a location of a particular gene. So you can see that this individual has a pink and a red, a blue and a red, this one has two blues, and so on. So I tend to think of these sticks. Mark thinks of the mice, I think of the sticks. Here's an intercross where you get mixing on both chromosomes going on. It's a little more complicated mathematics to keep track of. So we end up with some sort of a phenotype and trying to disentangle the variation in here. Part of the variation here is going to be just due to the random variation that has to do with microclimate and all sorts of things that happen to an individual, and part of it has to do with genetics. So we want to sort those two things out. And the way we sort that out is to have markers across the genome, and at each marker, so the markers are now across the bottom here, and these are individuals. For each individual you have these markers. I'm not going to go through the details of this, but the idea is that we have markers that are spread across the genome that are physically close to some genomic region, which we call a quantitative trait loci, or QTL. And that QTL is somehow causal for the phenotype that you actually measure. Okay. And there may be other covariates like sex or age or the things that come into play. Okay. And then for a particular marker, we may we measure our gross phenotype, like glucose level, and we might see a difference. Here we see a marker with a substantial difference in mean. Here, and on the right is one where there's very little difference in mean. And taking these, and then at each of these markers across the whole genome, we do a t-test. Okay, so Mark mentioned that last time. Okay, so that's as much as I'm going to show of that particular talk, and now we'll go back to here. Okay, so that's a 5-minute introduction to gene mapping. And but we're not looking at one measurement. We're looking at a bunch. So this is chromosome 17, and we're seeing a measurement here of insulin, I forget if that's glucose or insulin. This is our insulin turnover. So this is a gross phenotype on the top. I'm having trouble getting my mouse. There it is. Okay, so this is a clinical phenotype. And then these are all expression phenotypes down here, which seem to show a signal that somehow is related to that. And a start is just seeing that they're showing strong effects in this area. You can think of this lot as a minus log 10 of a p-value if you want. It's very similar to that. Okay, so we got a bunch of these coming up, and we might think, well, some of these are related, but which ones? How do we sort that out? So that's what we're going to be spending some time talking about. You saw this figure on the left yesterday from Mark Keller. On the right is just a count across the genome for each of these different tissues of how many hits we have, how many QTLs we have across all the expressions. So we got 40,000 expression traits. Every gene has one or maybe a couple probes in there, and you see we're having hundreds or even thousands of hits on a particular location. Some positions having hits across multiple tissues. Okay, and then this middle picture here is the one that Mark showed before. So now we're looking at a single chromosome and multiple tissues. The gray in the background is a SNPs. You might think there'd be a relationship, but there isn't necessarily. Okay. Now, one of the things that we're going to go through in a couple different stories here through today is taking these hotspots and trying to make sense of them. Okay. So the first part is what's a hotspot, and that'll be my second talk. I mean, how do we decide that something is a real hotspot as opposed to just a spurious relationship? So after lunch, we'll have a talk about that. And then Elias will talk about what do you do if you decide on some set, and I'll go through an example with yeast. This is a mouse example from the APOE from Jake Luce's lab, where we have here on chromosome 2, 80 traits that map on chromosome 2, and then we're looking at a subset of 12 traits which seem to be related in some causal way that we'll talk about. We can take those. One thing we can do is take those and organize them into a causal network, and Elias will talk about this kind of causal network. So I'm just showing it here as a taste of what's coming up. Okay, so that's sort of putting things in context. Now I want to step back from that and say, what is the nature of correlated traits, and try to look at it in a simpler way than trying to give you a little intuition about correlated traits. So why are traits correlated? Well, we're thinking about genomic regions important. So if we have two traits that are correlated with each other, then either one of them causes another, or else they have some sort of something that relates them together. One possibility is that there's one gene which affects both traits. That's called pleiotropy. You get multiple things affected by it. Another possibility is that there are two genes. They're closely linked. They each affect the traits, but what you're seeing is a correlation because they're linked. That is, if you have two locations on the genome, and you go back and think about this back through here. Okay, if you take two locations on the genome which are close together, they're likely to have the same genotype, whereas ones that are farther apart are more likely to be some rearrangement. So that's called linkage. Okay, back from the current slide. Okay. I'll talk a little bit about analysis of covariance as a way to think about that. If you don't know what that is, I'm just going to show pictures about it. There's more classical, fancier stuff that I'm not going to go into here. So why do we think about multiple traits and co-mapping multiple traits? Well, one reason is that we're just not clever enough to figure out what to measure. Okay, we're trying to measure something about why mice are diabetic or why people are diabetic. If we knew the right thing to measure, we could just measure that, okay? And we'd be done. But then we would, go from there directly to clinical trials. So we don't do that because we don't know what to measure. Another reason is it may be too expensive to measure that one thing. So instead, we measure 50 things, or 100 things, or 10,000 things, or 100,000 things that are somehow related to we hope are related to the thing we're interested in. Okay. And another reason besides not being able to measure the right thing is that there may not be one thing to measure. That's a kind of a reductionist approach. There may be multiple facets to what's going on. And we'd like to understand that whole situation. You saw several examples in the last few days from Gary and from Steve and Jeremy. And then we want to be able to separate out several things that are interrelated. So what we want to do is not look at things individually, but look at them together. So that's called co-mapping, or dealing with the multivariate nature of things. Okay. So I just mentioned that there are two types of of data that come up. One is where you have multiple traits on the same individual, and I think that's what we're thinking about a lot in this setting. But you can also have situations where you have multiple traits on different individuals. Classic one we deal with all the time is male versus female differences. So keep that in mind. Okay. So if you have, so here we have two traits called X and Y, okay? And these are characteristics of how they might be related. So there's a correlation here, a negative correlation between these two traits, but it's induced by the fact that there are two groups, okay? And in fact, the example that was brought up in the last discussion about the lean versus obese mice, the two different groups. If you put those two groups together and you start looking at correlation, any expression traits that are related to weight will show correlation just due to the fact that you've got lean mice and obese mice together. Okay, so here you have a spurious correlation due to the difference of the groups. Once you identify the groups, there's no correlation. And here we have a situation where there's no apparent correlation, but once you identify the different groups, in this case genetic groups say, then you see a strong correlation. And here we have both in play. We have a negative correlation that's due to these different groups and a positive correlation within each group. And so the challenge is to try to separate these two things out. And you can't do that unless you measure them together. Okay, so I'm going to talk now about a particular example. Brassica napus, it's my first exposure to QTLs. And we had two traits of interest there. One was 4 weeks and one was 8 weeks fertilization. So this is rapeseed or canola oil. And one of these is an annual, one of them is a biennial. But so if you, the biennial has to be vernalized, that is, you have to put it in a cold room in order to have it flower. So, and there's some specifics about the test. Let me just show you the data here. So here we've got 4 weeks, and we put on a log scale just so that things are nicely spread out. 4 week versus 8 week data. They're correlated with each other, and we look on a particular chromosome, and they're both mapping to the same place. They have pretty strong signals. They have slightly different characteristics about them. So this is the 4 week, and the dash is the 8 week. So we might say it looks like there's something going on there. Okay, so let's now try to sort that out using simple statistical methods. Okay, and what I've got here is I'm just taking this bottom curve and I'm reproducing it here. So the scale's changed a bit. And then what I've done is I've said, suppose that we first remove, if I'm looking at the 8 week, we first remove the 4 week effect. So we regress the 8 week on the 4 week and we take the residuals. Okay. Then what we see is this curve on the bottom. Okay. We've removed all the signal, apparently. Okay. Now, I'm not totally satisfied with that because it may be that the relationship depends on the genetic position. And there seem to be two humps here, and there was only one hump in the 4 week data. There's only one hump here. I've got two humps here, and I've removed both of them. So what if I allow some interaction? That is, depending on the locus, I could have a different relationship between the 8 week and the 4 week. Okay. And then you get this dash curve, which shows that there's still something there for the 8 week that isn't picked up from the 4 week. Okay. And that sort of makes sense. If you look back here, we have this strong peak at around 45 for this one group, and we have these apparent two peaks here. And it turns out there are two peaks. They've actually taken this to identifying the genes underlying these, and it's gone on from there. Okay, so now if we just go back to the raw data and just look at the data, and I think Steve was trying to emphasize this in a couple different ways, that you can get real fancy with this stuff, and you can fool yourself, and you might even get published, but are you going to understand what's going on? So if you can look at it in a simple way, that can be really helpful. So what I've got here is I've taken the marker at 47 centimorgans, and I've identified, this is like a back cross, there's two possibilities here. I've identified gray X's and black crosses for individuals, whether they were more like the stellar or the major parent type. And you see there's two different regression lines here. Whereas if I go at the 80 centimorgan position, there's basically one line. Okay. So this is some intuition behind this picture here, this curve here, that at this 47 centimorgans, we see something going on, and we don't see it going on here because basically the 8-week is tracking the 4-week with respect to that particular genetic signal. Okay, so we got two genetic signals. They're having different effects for the 4-week and the 8-week in one case and not in the other case. Okay, that just helps me sort things out a little bit using very simple plotting and messing around techniques. So we'll get to sophisticated stuff. Don't worry, Steve. Okay, so now multi-trait mapping. So we got multiple traits. We want to map them together, and we're going to have two curves sort of marching along, but they're correlated, and there may be something else going on there. So here we have a made-up example. This is a great paper, but it's hard to read, from Jiang Zeng. And he made up 3 different traits that have genetic correlation and environmental correlation. It's in an F2. And so there are 27 different possible phenotypes. And so what I've done is shown each of the subgroups, and the size of the subgroup is indicated by the size of the circle. And then the angle of the circle has to do with the genetic correlation between the two traits. And then you see that has to do with the environmental correlation. And the genetic correlation comes through looking across all these different circles. Okay. And then there's some magic that you can do to map these and try to sort out what's going on here. So what they're doing is mapping each trait individually. So these dashed and dotted lines are mapping each trait individually. And then this is mapping both traits together jointly and trying to ask the question, is there pleiotropy or close linkage? It turns out at this spot there's pleiotropy. There's one genetic signal, or one QTL, that's affecting both traits. Whereas over here there are actually 2 signals, and they're closely linked to each other. They're separated by 10 centimorgans or so, which would correspond to about a 10% recombination or 10% difference in the genotypes between those 2 positions. And the way you sort this out basically is to take these curves. These are individual curves, but if you think about looking at 2 together, you have the genetic signal, the t-test for the pair of them. You have a 2-by-2 situation. You take any 2 positions. So take this position and this position. You have 2 different markers. You can take all your individuals, divide them up into 4 different possible combinations. And then you can look at those pair of loci and ask if for one trait versus the other, do they have basically the same signal? And if they do have the same signal, then you should see basically what we're seeing over here. This is marching along the ridge of this 2-dimensional sheet to find where the signal is. And over here, the best position is actually off of the diagonal. I know that's kind of complicated. I won't. I'm not going to try to dig myself out of that one. But basically, there are statistical ways to sort out whether you have one genetic signal or 2 closely linked genetic signals, and that becomes very important. And it becomes particularly important when we start looking at thousands of traits that all seem to map to the same position. So do we have one signal cascade that's affecting lots of different things, or could we have multiple related cascades? I'm going to skip over that other part here. Okay, so now reducing many phenotypes to one. So sometimes it happens, and for the that you have things that are highly correlated, but you don't have one way to measure it. Here we have something about a shape, and I'll show you the shape in a moment. So we've got two different species of Drosophila, and we're looking at a part of the reproductive system, which is diverged between these two species. But we don't have an easy way to characterize that shape. So what they did was measure, they traced it basically and used some time series approaches, a Fourier series, to come up with 25 or 50 different numbers, and then they combine those together using principal components. So what is principal components? So you've heard about principal components a little bit in terms of looking at modules and getting the module eigengene is basically a principal component, as I understand it. So what's going on? If we have two things that are closely related to each other, then they're going to be correlated. And you can think of having this major blue axis here, where most of the variation is, and then another axis which is perpendicular to it, which gets at the minor axis. Of course, there's more axes, and you got to come up with more names. So if you take this whole thing and you just rotate it, in this case for about 45 degrees, then you end up with two new traits, PC1 and PC2 here, which now capture the same information as you had originally, but you now have two traits that are uncorrelated with each other. Okay, so if they're uncorrelated with each other, we don't have to worry about trying to, so we don't know whether it's genetic or environmental correlation, but we can look at them separately and assess each of them on their own. Whereas on the right-hand situation, if I map this guy and then I map this guy, I'm going to be telling the same story twice. And as you see here, 93% of the variation is in this one axis. Most of the information is along this axis. So that's where we want to capitalize on. Okay, so in the case of the Drosophila, they're looking at shape. Here we have a beautiful separation. I've never seen anything like this. So here's the shape of this reproductive piece, and reconstructed from 25 Fourier components, from 10, from 3, from 1. The first one is basically getting the size. The second one gets a rough shape, and so on from there. But when you combine those together, get the principal components of these 25, the major axis, it explains 80% of the variation. And here's the next, between these two, you get 90% of the variation. There's basically nothing else to be said from these. These aren't the first two principal components, the first two Fourier components, but they are some weighted average of all these components. Okay. And what's neat here is that you've got the parents are basically at the end. The black triangles and the black squares are at the end. The F1's in the middle, and then these are back crosses in either direction, and they're nicely spread out. Okay. So now there's one measurement for shape. We didn't know how to characterize it. We made lots of measurements, came up with some sort of nice combination. And then always good to check. Does this mean anything? Does it make sense in terms of what you're looking at? So here we have the original shapes that are ordered basically by this measurement, by this one axis, and seems to do a pretty good job of capturing what your eye captures. That's one thing you want to try to do. Okay. And then these are just some ways to do gene mapping. It's just showing that in this situation, it's fairly complicated. They picked out 20 different QTLs, 20 different genomic regions that seem to be related to this trait using various different methods. This was a crude earlier method called CIM, which is no longer recommended. This is basically looking at trying to say there may be multiple genomic regions that are affecting things, so let's look for multiple QTLs at the same time. And this is another approach that I was involved with, the Bayesian interval mapping. I'm not going to talk about that. Okay, so what have we got going with the multi-trait strategy? Okay, so what are we trying to do in this setting? And we'll carry on through the day on some aspects of this. So first, use some data reduction method. You can just try principal components on its own. Doesn't work with expression data. You got to be clever, and one of the clever things here is this WGCNA, and then principal components after you've done that, not on everything together, or some kind of clustering. Well, this is using clustering. I'll be talking in the next talk about hotspots. We got a bunch of things that map to the same location. They might correspond to modules, they might not. It's coming at it from a different angle. And then another approach that you've heard a lot about, but not without the genetics, is using functional or pathway information to try to organize things. So you're trying to reduce, organize into groups that make sense. Okay. And then gene set enrichment's been brought up, and another thing that Mark focused on a bit is correlating with key clinical traits. Okay. So we're going to talk about using genetics to fine-tune the search, and you heard a little bit from Mark, you heard more from Elias this afternoon. And then there's other fine-tuning that various groups do in terms of looking at SNPs and identity by descent to try to, because as Mark was pointing out, I think he pointed out yesterday, that we have this great region we're interested in, but it's got 300 genes in it. So you've got to come up with other ways to screen those out further. Once you've done some sort of screening and got down to a finer scale set, then we can start talking about building causal networks, and Elias will talk about that, where you're trying to get at the underlying pathways using the data you have in hand. And you might also want to use biological information. We won't talk about that very much. And you've heard about hairballs. And then validation tests is, of course, what you want at the end. So, let's see, how are we doing on time here? We're getting right about on time. Okay, so so when we talk about functional information, you've already heard various things about this, but I just want to remind you of a distinction that was made by a couple different people, that some of the information you have in databases provides direction and some doesn't. So gene ontology and KEGG may or may not give you a direction. Well, gene ontology generally doesn't. Knockout panels will often give you a direction. You knock out one gene, it affects other things further down some signal cascade. Protein-protein interactions typically will not give you direction. Transcription factors will typically have direction associated with it. There are methods that only use this information. You've heard a little bit about some of that. But one of the things we've been exploring, and we have a paper in a book chapter, which you can find from my website, but we're not going to talk about it in detail here, is using that information to improve on causal networks. Okay, and this work is joint work with quite a number of people. You saw pictures of all these people yesterday, and there's a variety of support for this. So I think this is probably about a good time for questions. I have other stuff that I can go into, but I think it would be better to wait till after lunch, the next talk. Yeah. You all ready for lunch? Yeah.
So is there a good R function for distinguishing close linkage versus pleiotropy? Or what software tools would you use for that?
Well, so in RQTL, if you look at Broman's book, Broman and Sen's book, there's a whole chapter on multiple QTL. And there's a number of tools that they have available. So in addition, Jiang Zeng has incorporated a number of his ideas into they have a standalone package called QTL Cart from North Carolina State University. So just look up QTL Cart, you can find that. It's a Windows-based, PC-based system. But a number of those ideas are now incorporated into RQTL. Well, they work well enough, but the problem is when you're working with inbred strains, as I'm talking about here, and as Mark was talking about yesterday, you have a problem called linkage disequilibrium, that you have hundreds of individuals, but when you can only narrow down a region to, say, hundreds of genes, and Mark can only test 3 genes. So I can't give him, if I use these QTL methods on however many traits I've got, I'm still going to end up with a large region. I mean, it may look small, but then you go drilling in for genes, and going from a QTL to a gene is a tough thing. And that's a limitation not of the statistics, it's a limitation of the nature of the experiment. So if you want to get down to fine mapping, you either have to go to advanced lines, or you have to go to mutation or some other system. Yeah.
The experiments what you have shown is using the EQTL experiments what you have shown here is mostly the linkage-based analysis.
Yes.
Not the association tests you are doing here.
I'm not talking about association tests, although many of the methods can apply over there. There's a complication though, because when you get into association, you also have problems of admixture. You have questions of whether what you're seeing has to do with the historical development of that population you're studying. So you look at a natural population, there are certain selection that's gone on for that natural population. Once you do an artificial cross, you break up that.
Currently there is a big interest in doing EQTL analysis in human subjects.
Yes.
Especially the expression QTLs in different tissues.
Right.
And people are using the expression-based test, and they are trying to remove all those residual effects, including the admixture, genetic admixture.
Yeah, they're trying. There are not many successes yet. So it's very hard to do that. The other challenge is that you have many alleles to work with, and some of those alleles are very rare. So in experimental cross, you can control the frequency. So another direction that's going on are things like the collaborative cross and the diversity outcross in mice, and there's also a panel in Arabidopsis where you start from multiple, say 8 different inbred lines, and then you manage the development of a population. So you have the heterogeneity that you see in a larger population, but you can control some of these issues of selection bias to a certain degree, because there's still going to be selection in terms of who survives down through the line. But yeah, you can take these ideas, carry them over into outbred populations, and people have done that. Yeah. Okay.
Thank you very much, and we resume at 1:30.